In this video, we are discussing structural aspects of defense system of lung. Lung is exposed continuously to inhaled particles and microorganisms. To counter this, defense mechanisms are embedded within anatomical structure of respiratory tract, extending from upper airways to alveoli. Anatomical levels of structural defense. Structural defense system of lung is organized at multiple anatomical levels, including upper airways, conducting airways, alveolar region, and the interstitial and lymphatic compartments. Upper airway structural barriers. In upper airways, nasal vibrissae and turbulent airflow act as a structural barrier that trap large inhaled particles before they even reach lower respiratory tract. Structural defense by conducting airway epithelium. Conducting airways are lined by pseudostratified ciliated epithelium. Tight intercellular junctions form a continuous physical barrier that prevents direct penetration of inhaled pathogens. Defense by mucus layer. A continuous mucus gel layer covers the airway epithelium. This layer traps inhaled particles and microorganisms, preventing their direct contact with epithelial surface. Role of globate cells and submucosal glands. Globate cells within airway epithelium and submucosal glands within airway wall serve as a structural source of mucus in conducting airways. Cilia as a mechanical defense structure. Cilia form a dense, structurally organized lining on airway epithelial cells. Their uniform orientation towards the pharynx depend on integrity of airway epithelium. Bronchial associated lymphoid tissue. Bronchial associated lymphoid tissue consists of organized lymphoid aggregates located within airway wall. These structures are more prominent during chronic inflammations and are a part of airway defense structure. Structural changes in distal lung. In distal lung, cilia and mucus are absent. Structural defense in this region relies primarily on cellular components of alveolar wall and air spaces. Alveolar epithelium. Alveolar epithelium consists of type 1 and type 2 alveolar cells forming a continuous cell lining that separates air spaces from interstitium. Alveolar macrophages. Alveolar macrophages are present within alveolar spaces and along epithelial surface, forming primary cellular defense component of distal lung. Interalveolar septum. Interalveolar septum contains pulmonary capillaries and interstitial immune cells and plays an important role in structurally containing the inflammatory processes. Pulmonary capillary endothelium. Pulmonary capillary endothelium forms a continuous barrier separating blood from alveolar spaces and regulates cellular movement across the alveolar wall. Pulmonary lymphatic system. Pulmonary lymphatics are located within interstitium and contribute structurally to defense by draining excess fluid and clearing particulate material. Connective tissue framework. The connective tissue framework composed of collagen and elastic fibers. They provides mechanical stability and limits spread of injury within lung. Structural compartmentalization of defense. Lung defense system is structurally compartmentalized into airway, alveolar, and interstitial compartments, allowing localized containment of injury. How does structural failure of defense occurs? Disruption of epithelial integrity, loss of mucociliary interface, or damage to interalveolar septa all can lead to failure of structural defense mechanisms. In summary, the lung possesses multiple structural defense mechanisms involving airway barriers, alveolar cellular components, interstitium, and lymphatics, all of which are essential for maintaining pulmonary integrity. This diagram summarizes the structural defense system of lung. In conducting airways, ciliated epithelium, mucus, globate cells, submucosal glands, and bronchial associated lymphoid tissue form the primary physical and immunological barrier. In the distal lung, defense is provided by the alveolar epithelium, alveolar macrophages, interstitial immune cells, capillary endothelium, and lymphatics, which together limit particle penetration, contain inflammation, and preserve alveolar integrity.